Hi, my name is Bob Makinson. I'm a conservation botanist based in Sydney, and this is a brief introduction to myrtle rust. It's based on a presentation given on 22nd of February 2024 to a horticultural practitioners forum hosted by the Botanic Gardens Australia and New Zealand and the Australian Network for Plant Conservation. It's a very quick summary of myrtle rust in just 8 slides. And apologies to overseas viewers. This deals very much in a summary way with the Australian situation. Myrtle rust is a vast problem with a big literature, and it's become a global biosecurity and plant health problem. So there's a lot going on overseas, which I won't even attempt to cover in this talk. Just loading up the presentation. So myrtle rust is an introduced exotic fungal disease. It's originally from South America. It manifests most usually as yellow powdery pustules and spots, as you can see in the photo at top right there. It attacks only plants in the Myrtaceae family. So if you've got a plant that you are sure is in that family and you're getting a rust-like disease that does manifest as these yellow pustules and powders, it's quite likely that you're dealing with myrtle rust. If, on the other hand, your plant is not in the Myrtaceae family, then it's not going to be myrtle rust. It may well be one of the other yellow rusts, of which there are many. Frangipani rust being an example. Myrtle rust likes moister habitats. It is not a problem in inland Australia, except in very protected situations like green life industry, nurseries and things of that sort. Myrtle rust spreads very easily. The fungal spores are airborne and extremely lightweight and very easily transported either by wind, animals or humans. It has a rapid life cycle, just 10 to 12 days from one spore to potentially thousands from one point of infection. And it has a huge host range of species within the Myrtaceae family, over 500 species globally, and that's going up all the time across 69 genera. Most species do vary in susceptibility. There are a few species of Myrtaceae that don't seem to support the rust at all, but most do. And the level of susceptibility varies from very low to very, very high, to the point where there's a significant number of species in the extremely vulnerable category, and those are the ones we are most worried about from a conservation point of view. Now, multiple strains or variants of this pathogen are known from various countries. There is only one variant in Australia so far. You'll hear this referred to as the pandemic strain, and it's a national biosecurity problem, or a national biosecurity priority, I should say, to keep out the other strains because they could add to the very significant problem that we've already got. One of those strains in South America is particularly aggressive on eucalypts in the industry there, and forestry and timber products industry. And we certainly don't want to add to our burden of threat in Australia by introducing that particular variant or any other. Now, myrtle rust can complete a short version of its life cycle, an asexual version, on any host species, any species of Myrtaceae which can support it. It doesn't need an alternative host like some rusts do. It's fairly sneaky in the way it operates. It attacks new growth, not older leaves, just new shoots and leaves, and occasionally, in some species at least, flowers and fruits. And basically, highly susceptible plants cannot renew their foliage. There's often no flower or fruit set because the new shoots have been killed that would have supported those. So you get death of the individual plant, and at a species scale, you may get local or total declines and extinction. Myrtle rust, as I say, originated in South America, basically the Brazil, Uruguay area. It got into the Caribbean islands in the early part of the 20th century and decimated the allspice industry there. It got into Florida in 1977, where it did very well on introduced Australian broadleaf paperbarks growing in the Everglades. It got to California and Hawaii in 2005, and from somewhere, we don't know how, it got to the Sydney region of Australia in early 2010 or a bit before. It has also spread to numerous other sites in the Indo-Pacific. In Australia, myrtle rust is now thoroughly naturalised along almost the entire east coast, from Cape York right down almost to the Victorian border. And it's present in Victoria and Tasmania, but so far only recorded in cultivation in the green life industry or on outdoor cultivated plants. It's not been recorded in the bush on wild plants in those two states. It's entirely possible that it is present. In fact, it's probable that it is present in the wild to some extent, but just not manifesting very strongly down there. And that's probably partly climate, but also partly a lack of the highly susceptible species, of which there are many more further north. In the Northern Territory, there's a scattering of records right across the top end, and it's pretty well established there, but the level of monitoring is pretty low, so we don't really have a comprehensive picture of what's happening there. It did creep across the border into the far north of Western Australia in 2022 on paperbarks, but so far has not broken out of that region. Keeping myrtle rust out of South Australia and particularly out of the southwest of Western Australia is a domestic national biosecurity priority. In the far southwest of WA, in the regions that are thought to be most capable of supporting myrtle rust climatically, there are well over 1,000 species of Myrtaceae, many of which are known from screening tests elsewhere to be very susceptible to infection, and it would be a biodiversity disaster if myrtle rust gets there. We don't know how myrtle rust got to Australia. It could possibly have been by wind. Some rusts have been recorded as travelling intercontinental distances, but it's probably much more likely that it was brought in either by illegal importation of plant material or on somebody's clothing or gear returning from overseas. This set of slides illustrates just how easily one can become a vector for it. This was a street tree of Syzygium jambos rose apple in Brisbane some years ago, which you can see the new growth is thoroughly rust infected. It's just got pustules, yellow pustules all over it. And you can see the close-up there. They are shedding thousands and thousands of spores, and you can see the ruptured pustule at bottom left. Now, brushing past that tree with a polar fleece on, you can see that one can pick up a very large load of spore, and that will stay viable for an uncertain length of time, but potentially weeks. And if you are then visiting somebody's nursery of prize Myrtaceae, or if you are travelling to Western Australia or travelling elsewhere to a sensitive area that hasn't already got this particular strain of myrtle rust, you could very well be a carrier. Now, fortunately, it is easy to delouse yourself in this kind of situation. And if you are travelling from the East Coast to WA, particularly if you're going into the bush, but it's a good idea for any sort of trip from the East Coast to WA, launder all your clothing, including hats, in ordinary washing machine cycles with ordinary detergent immediately before you go. Don't wear them into the bush. Don't take anything that you've worn in the bush that hasn't been thoroughly laundered. Washing machine detergent on a regular cycle will kill the spores. For things that can't easily be laundered in that way, like tarpaulins, tents, sleeping bags, some hats, camera cases, things of that sort. Either a thorough wash down with a solution of a strong industrial detergent, truck cleanse or something similar, or a spray of 70% methylated spirits in 30% water will kill the spores effectively. But they do last for weeks. You do not want to be the vector that takes this to anywhere that hasn't already got it. So I said earlier that myrtle rust was a sneaky sort of disease because it attacks new growth. You can see the top left shots here, a couple of levels of infection on new foliage with yellow pustules and the darker areas of dying leaf tissue around them. At bottom left, you can see some suckers of native guava, Rhodomyrtus psidioides, which have emerged. The original adult tree was killed by myrtle rust, but the root system is still active and pushing up these suckers. And the myrtle rust does attack them as soon as conditions for it are opportune to do so. In this particular case, that's the shot at bottom left, you can see that the sucker has managed to push a few leaves through to maturity, and they do not get infected, and they are still performing a photosynthetic function that may get that root system through another year. We've now got root systems in that particular species, native guava, that have been throwing up these sucker fields for 14 years, even though the parent trees are dead. But sooner or later, they're going to give out. The strain, the pandemic strain of the disease that we've got in Australia at the moment, infects many species of eucalypts, but mostly not terribly aggressively, but occasionally you will see it doing some damage, and there's a shot there of Eucalyptus tyndallii, some coppice growth being hit pretty hard. Next to that is a shot of scrub turpentine, Rhodomyrtus rubescens, a fruit, one of the rare fruits that has managed to set, because the shoots on which the flowers occur are usually killed outright, but a few get through. But then the fruit itself gets infected, and there are no reliable cases yet of finding viable seed in fruits in the wild, or very few. One of the scariest photos I've seen in 14 years is the shot of the eucalyptus plant Choniana cotyledonary seedling at bottom right. You can see faintly the yellow spots on that seedling. And of course, if that's happening out in the bush at such an early stage of growth, it's unlikely to be observed except by the most sharp-eyed of people. And probably the first thing we'll know about decline in a species where that is happening, if the adult plants are a bit more resistant, is that there will be suddenly an age gap in 15 or 20 years with no younger plants coming on. The mode of action, the most common mode of action for myrtle rust is shown by the shot at top right, which again is scrub turpentine, Rhodomyrtus rubescens, north of Sydney. This shot was taken in March 2011, and prior to that, in December 2010, the plant had put on its normal new season growth, which had been killed pretty quickly by myrtle rust. And those are the 4 dark brown shoots that you can see, one of them with a dark brown shriveled leaf on it. They've been killed outright by myrtle rust in December, January. In autumn, the plant put on a second flush, an autumn flush of growth, some new shoots, and those in turn, these are the lighter coloured ones, those in turn are now being attacked and killed by myrtle rust. The older leaves, the mature leaves from previous years, as you can see, are unaffected, but of course they won't last more than another year or two. So the net effect is that this plant has put on no new foliage in that year, and as the older leaves die, the plant will become progressively defoliated and will starve to death. Now, there are treatments for myrtle rust in cultivation. They're, for various reasons, not terribly satisfactory, and they're not by and large deployable in the wild, except at very small scales. Fungicides are effective, but they need to be used carefully and in rotation. They are usually too toxic to other organisms and too broad spectrum in their action on fungi for deployment in the wild, except on individual plants, either for experimental purposes or if there's a particularly iconic or sacred individual plant which has to be protected at all costs. So fungicides are feasible. Rotational recommendations and permit situation varies from jurisdiction to jurisdiction, so check with your local Department of Agriculture or primary industry as to what is recommended in your state, and follow the label advice carefully. The question of bacterial or other biocontrols, parasitic fungi that might eat myrtle rust, things of that sort, is often raised. Some such things do exist. They are known, but not well, and their effect seems to be generally fairly weak. Delivery of them would be fairly difficult. Their action is probably not fast enough to keep up with the potentially very rapid proliferation of myrtle rust and its reproductive cycle. And of course, there are ecological issues with deploying any improved strains of such things because they might also attack other more beneficial fungi, and we don't want to do that. There is some interest overseas and a little bit in Australia in the possible role of endophytes, that is microbiota that occur inside the host plant, and their role in helping the plant to resist diseases of various sorts. There is some evidence that particular balances of endophytes can have an effect on myrtle rust levels of infection and severity. And there's some exploratory work underway in New Zealand. But again, manipulating that sort of situation in the wild is extremely unlikely. There's a recent development in Australia of a spray treatment based on double-stranded RNA, interference RNA. It's still under development, but basically it's a means of interfering with the metabolic and reproductive cycle of the Myrtle Rust pathogen. It's very specific to that pathogen. It doesn't attack anything else. It's non-toxic, and it doesn't attack anything else. It's like a lock and key situation where the molecular structure of the RNA can only key onto this particular pathogen and not onto anything else. Now, it's early days for this. It's still under development. It's potentially applicable to a bunch of other diseases as well. But the developers at the University of Queensland and a consortium around them have focused on myrtle rust initially. Whether it goes through to commercial scale production and deployment, and if so, whether that will be for myrtle rust or for some other disease, is an open question at this point. Getting these things to market is often a major issue. It's likely, if it does become a reality, it's likely to be a big help in cultivation, including in the protection of conservation collections that are held ex situ, that is, in cultivated situations, not in the wild. But it's unlikely that it could be deployed in the wild, except, again, as with the fungicides, at a very, very local and very limited scale. We simply can't spray the entire East Coast every few months. The persistence of the RNA interference spray is also a bit uncertain at this stage. It's of the order of weeks at this point. Whether that can be extended at all is an open question. So there are treatments for cultivation, but not in the wild by and large. So for conservation and species recovery, we really need a large, dispersed, strongly coordinated and protected collection with as many wild genotypes as possible, as much variation as there is in the wild, if we can possibly manage it. And we need that because if we are to screen for possible resistance traits, we need a big library of material to draw on. The resistance may be rare, as it is in native guava. We haven't found any yet, but it's probably out there, but it may need amplification and sort of waking up for it to become an effective trait. We also need a big library of ex situ material, living material, because for eventual reintroduction, we don't just want to put back clones. South Americans have been breeding for resistant eucalypts for decades now, quite successfully, for another variant of this disease. But they're interested in clones, or at best land races with minimal variation. For conservation purposes, we're interested in preserving the full range of adaptive variation that exists in a wild species, but simply adding to it a greater capacity for tolerance of this particular disease, enough to get it through the threat that is posed to it. So the requirement for quite large collections is really a major logistical issue for us. But the key questions with this approach, of course, are: do resistant genotypes exist? Can they be selected and bred into a species without bottlenecking the variation? And can a wild species then be reintroduced or wild populations reinforced? Now, I haven't got time in this talk to go into it, but the answers are yes, yes, and yes. The technical obstacles to this strategy are all solvable. The question is investment and will, if you like, as to whether we are going to do it and protect our species. So the vision for species recovery looks something like this. For the most severely affected species, we have to secure as much of the wild germplasm as we can accommodate quite quickly. The window of opportunity for some species is closing extremely rapidly. They simply won't be there. They will be effectively extinct or too hard to find in the wild very, very soon. We've begun this process with about 4 species so far, but there are probably between 30 and 50 species for which it needs to be done. And while we're doing that, we've got to try and establish a project infrastructure with the resourcing and collaborations that go along with that, and get this germplasm material into safe custody and begin the process of screening to identify resistant genotypes and eventually to start selecting those, breeding for them, and infusing those traits back into the full range of variation of the species. And while all that's going on, we have to secure the long-term resources and get a social consensus around this recovery strategy before we can begin the process of reinforcing surviving wild populations or reintroducing in the case of species that have gone extinct in the wild in the meantime. This strategy is outlined in several documents now, but 2 of them that are easily accessible are a review of Myrtle Rust from 2018. Things have moved on a little bit since then, but the basics of the plan, or the basics of the approach, are laid out there. And the more recent 2020 Myrtle Rust National Action Plan, you can get both of those from the ANPC's Myrtle Rust pages on our website. So most of us in the horticultural space are really in that central circle, dealing with the safe custody issues, getting the material in from the wild, establishing meta collections dispersed to multiple sites for safety, and beginning the process of finding the resources to move to selection and breeding. An example, one of 4 which are really solidly underway, and a few more which are just barely beginning at this point, But one of the more advanced cases is native guava, Rhodomyrtus psidioides, where the wild sampling and ex situ collection development has been fairly advanced. And these shots are from the New South Wales part of that effort, which is focused in many respects at the Australian Botanic Garden, Mount Annan. I mentioned earlier that native guava does sucker. Now we can take aerial cuttings from that, as in the centre shot in the top row here, or we can take actual sucker units off the spreading lateral roots. These are the suckers that pop up every year. They eventually get killed by myrtle rust, but in the meantime, they help keep the system ticking over. And those sucker units are actually faster and really robust. They go like a clappers. So they're great propagation units. On the other hand, we don't want to exhaust those root systems. So a bit of care is needed, a bit of balance is needed. But whether it's suckers or ordinary aerial cuttings, the plants are relatively easy to establish in cultivation. And in pot, even, they are quite precocious. They flower and fruit in their second year if all conditions are right. That's a good thing because it means we can begin to develop both breeding and screening and seed capabilities very quickly. So there's been work going on germinating the seed, which is not really terribly amenable to ordinary orthodox seed storage. It can be stored a bit, but not for terribly long. And we can begin to rear, either from cuttings or from seed, sets of plants that are suitable for screening purposes, or for dispersal as part of a permanent conservation meta collection to multiple sites. There's also some parallel work going on looking at tissue culture and cryopreservation in this and other species. It's quite labour intensive and quite expensive to do that, but of course it is a more compact form of long-term storage. But the reality is that for screening purposes and breeding, we do need a ready stock of large quantities of actual live emergent plants. So this is the kind of work that's going on now, as I say, for about 4 species, and we need to extend it to others. So I will finish there, but that's a brief outline of where we're at. Thank you very much.